So hello everyone, both here in the room and online. So we are happy to start this new cycle of the seminars in Ecology and Evolution of Montpellier. So as some might have already noticed, we are a new team that is organizing and coordinating these seminars starting from today. We would like, well, the seminars are still also with the support of Paul AEB and the PTL3 of the University of Montpellier. And before starting with the presentation, I would also like to thank the old committee for their work and dedication for the past few years. And with that, I'm going to give the floor to Anne-Nathalie Volkov to introduce today's speaker.
Okay, thanks. So I'm very pleased to introduce Vincent Doublet. So Vincent has done his PhD in Poitiers in 2010, and then he followed up with several postdocs in Germany, then in United Kingdoms, and finally got a teaching and research assistant position in the University of Ulm. And finally, he joined our lab this year in March. And so he's a evolutionary scientist, and he was hired in Montpellier to work on eco-immunology of insects. And so we are very pleased to have him joining our lab.
Good. Thanks, Anne-Nathalie, for this introduction, and thanks everyone for being here. So yes, I just arrived in Montpellier, so start to see who is working here and try to map all the different labs. And I'm really pleased since the last few months I spent here was really great. So yeah, I'm looking forward to meeting all of you and talking about insect and diseases, because that's what I'm doing. And today I'll be talking about bee diseases. I've been working on bees for the last few years. Let's say 12 years. And today I'll be presenting work I've been doing on disease transmission in the bee communities. And that's work that started in the UK at the University of Exeter, and that I continued developing at the University of Ulm, where I was teaching there. So I like to start by framing this work on a general context of emerging infectious diseases, which one of a major threat for global health and security today. And over the past decades, there have been a huge increase in the detection of new diseases and new outbreaks. And this is no coincidence, and we understand which factors actually have an impact there. And I can start with extensive urbanization that steps on natural habitats that will generate new interface between human populations and wildlife. I give here another example here: interaction between climate change and human population that also increase the viral sharing between wild animals and humans. And on top of that, you have a lot of other factors, including globalization, that will fuel this transmission and the spread of diseases in a large scale. And this awareness led to new projects, substantial international projects investing in surveillance and preparedness for new outbreaks. And here I mentioned two examples of multi-million international programs to follow new outbreaks, to identify the factors that influence these outbreaks. And also these outbreaks, and we know it now from COVID, it can have a huge impact on our societies, on our behavior. And just to recall how efficient was this mass change in behavior for social distancing to reduce transmission of disease and to protect the most vulnerable people in our communities. And this pattern of increasing outbreaks is not restricted to human societies. It's also what we see in plants and in wild animals. And global change, that is through either a shift in agricultural practices or in the changes in natural habitats, can also impact these different plants and animals. And here I give two examples that show how dramatic these outbreaks can have in societies. Here I give the examples of the Phytophthora infestans outbreaks that led to the Great Hunger in the 19th centuries in Ireland, that leads to massive movement of population towards the Americas. And another more recent example is the Cretidiomycosis of amphibians, that here had a huge impact on aquatic biodiversity. So on my side, I'm focusing on diseases in bees. And the reason I've been working for so long on bees is because these insects are indeed extremely important at different levels. First, they are really important for food productions. They are pollinators, are a huge majority of plants and pollinator of plants in the production of fruits and vegetables. And they are also very important for the maintenance of our ecosystems as pollinators of wildflowers. Yet bees are in trouble. You all heard about it. The bee industry is always experiencing losses of colonies, and the wild bees also are in decline, both in abundance and also in terms of diversity of bees within natural habitats. And here are two examples in Europe, in the Netherlands and the UK, where in the past years we've seen a reduced diversity of bees. And the reason for this decline is multifactorial. And we know that, for example, that for honeybees, mass beekeeping is a big constraint, or it's a big pressure on the health of the animals. There is also environmental impacts of extensive agriculture, including the use of agrochemicals, poor nutrition, and fragmentation of habitats that leads to lower access in forage, which means less diversity and abundance of flowers that the bees are foraging on for nectar and pollen. And on top of that, diseases, which include emerging diseases, especially for the honeybee, but also for wild bees, since globalization and the movement of honeybees. There have been a few diseases that have been a jumping host and traveling around. And in my work, I've been focusing on these different factors, and especially the interaction between them, looking at what are the factors that can influence the virulence of the different diseases, and also on the transmission. This is the second part I will talk about today, how these different factors can influence the movement of diseases within the bee community. So again, why working on bees? They are actually really relevant models to study movements of diseases. And here I'll be talking today about viruses. So the bee community is represent like a huge community of different hosts, and we can even include other pollinators, insect pollinators that we forage on the same resource. And we know that through the use of flowers, and especially the collection of nectar, they can spread diseases to the different members of that community. And what is interesting is that transmission opportunities are easy to quantify, as from plant pollinator networks, as just mentioned, through observation. But also we know that there is this mite, the varroa mite, which is a major vector of viral diseases in the honeybee. That is an invasive species that jumped from the Asian honeybee Apis cerana to the European honeybee around Korea and Russia. And since the movement of honeybee population in the world, now it's almost everywhere. Only tiny islands are devoid of varroa, and it's only like the last 2 years now that it's also in Australia, and it's invading this island very quickly. There is also a diversity of lifestyle from very social species like the bumblebees here and the honeybees to solitary bees, but also parasitic life of bees parasiting the nest of other bees. And as I mentioned, there are diseases that are transmitted by varroa that are extremely important economically. And here I'll mention, but I'll come back in more details later, the four-winged virus that have been associated to a high mortality over winter in Europe and in Americas. So I'm interested in this movement of diseases through the bee population, and for that I wanted to again work in this framework of understanding the mechanism of disease emergence, which generally follow these 3 steps. First, we need an exposure of a virus or a disease to a new host species or a new host population. But then the second step is infection. It doesn't happen all the time. But when there is a successful infection, then there is a possibility for transmission, and that's the last step. And when you follow these 3 steps, this is when disease emergence occurs. And if we know now from field work, we know quite well which viruses can be found in which pollinator species. We know much less about how this is transmitted, how often, and what are the opportunities of transmission between these different insects. And this is on this aspect that I've been working recently, using tools that emerge in epidemiology, which are like contact networks. Contact networks is basically looking at who is interacting with whom. So following these networks of contact between individuals, and superimposing data from disease presence, and that's when we can actually identify the factors that influence the transmission within a community of individuals. And that tool, which is widely used in epidemiology, is also more recently used in disease ecology of wildlife. And I was particularly inspired by this example of the bird feeder, where it has been shown that these bird feeders we use in our gardens can increase the chance of disease transmission of fungus that can transmit from bird to bird through the use of the same food resource. So with this example in mind, we had as an idea to work on what could be a bee feeder. And we came across this. These are wildflower strips. So you probably heard of wildflower strips. It's a conservation program to attract bees locally and in agricultural settings and to boost the population. So basically the farmers receive subsidies to use part of their fields, not for growing plant, but to grow wildflowers and attract pollinators. And it is good in terms of nutrition for a conservation program. It brings a lot of insect pollinators, and locally it can also help for pollination. And we know that it can increase yield on apple trees, for example. So it's good for nutrition. It brings a lot of insect on the same spot, but this is where we are asking the question, what's the impact of this practice on disease transmission and on the network of interaction between the different species in these fields? So to study this, we did a 1-year study in the south of England. So we visited 10 farms, so 5 control farms not involved in sowing flowers for bees, and 5 farms that were involved in this conservation program. And there we visited it 4 times within 12 months. And we collected data on the plant pollinator networks. So looking at which species were foraging on which flower. we're collecting data on flower diversity using quadrats, and we've collected more than 6,000 samples to identify the viruses that were present there. And we mainly targeted 30 individual of the 5 most common species that we could see on these flowers, which quite often, not always, but quite often, was social bees, of course, so bumblebees and honeybees. Social bees only because the foragers is a caste of workers, non-reproductors, and they can recruit each other when they find a good spot of food. So that's why we generally, when there is one bumblebee, the sisters are also around. So we had, that's why they were quite often there in high quantities. And also, this program is really targeted to attract bumblebees, which are very good pollinators as well. Right, so what I'll be talking about today, so I'll slice this work in 3 steps. And the first question was, what all the wildflower strips are doing on the plant pollinator network? Does it have an impact on the interaction between the different members of that community? The second, can this program be a hub for disease transmission? And here we use 2 focal viruses that we follow across the different individuals in our field samples, and then I will explain how the virome of the bees that we've collected, how is it shaped, what are the factors that are involved in shaping the viromes of bees in natural habitat. So I'll start with the how flower provision impact plant pollinator networks. So this is one of the site where we've been collecting data, and you can see how rich in flower it is. And basically, the first step was to make sure that this is indeed different and efficient in terms of flowers and in terms of bees visiting. So what we see here is that the wildflower strips are visited by, there is more flowers visited by insects compared to the other field margins where there was no flowers sown by the farmers. So we found more flowers in terms of diversity, but also in terms of density. So there was more resource in mass for the bees. And in the end, what we found is that in wildflower strips, compared to other field margins, showed more higher bee abundance than the random other field margins. So that's good. That means that it works, and we identify flower richness and flower density to be involved in the attraction of bees. So that was our first step. And then, given that it has an influence in bringing bees, we wanted to know whether it changes the networks. So the networks was working along the transits and recording which bee species is foraging on which plant. And in the end, what we did is translating that into a contact network, which is basically using indices like niche overlap and see who is sharing the same niche with whom. What we found here is that when flower species richness increases, we have lower connectance, which means that when there are bigger networks, we have less connection within the networks. And this is a classic, but that also means that when there is choice for the bees, they do tend to choose their flowers and not forage on everything. And that's also what we see here. So we have also it's on the edge of being significant, but we saw that flower species richness also had an impact on niche overlap, and meaning that here there is a lower niche overlap when there is more flowers. So again, they tend to separate their foraging niche when there is more flowers to choose in locally. So that's an interesting result. And that means that when we have more bees on these flower strips due to density and diversity, these factors can be manipulated to play on the network. And potentially with lower connectance and lower niche overlap, we could expect a potential impact on competition because they are not foraging on the same food. But here our interest would be also on disease transmission. And if they are not foraging on the same food or less often, potentially that also means that there will be less transmission across species. So that's what we wanted to check. So that's when I start talking about viruses in the second point, and link this agricultural practices to the presence and the abundance of the viruses in the bees. So here we decided to focus on two viruses, and we decided to go for two very different viruses. And the first one is the deforming virus. So that's a flaviridae that it's transmitted, where it's known in bees for a while, and it was like kind of a mild disease before the invasion of varroa mite. But since varroa mite arrived, a new transmission route was possible, and now it's directly injected into the hemolymph for the bee by the mite, and it becomes a major problem. And now it's linked to overwinter mortality of honeybee colonies. And in past work, we've been showing that even a new strain, the deforming virus B, that somehow is replacing type A or interacting through recombination, can lead to faster mortality of colonies. So here, this is simulation from a lab experiment. We can see that this higher mortality, this higher virulence of type B can lead to a shorter life of 1 year of the whole colony. And we also looked at the overall viral landscape when varroa arrived, and we showed that it can transmit a lot of different diseases, and the virome of the honeybees dramatically changed since the invasion of the varroa mite. So this is the first virus we looked at. So it's basically a huge problem for the honeybee. And we compared our data to another virus that were present in this community, which is acute bee paralysis virus. And this one is rather a bumblebee virus. It was described originally in honeybees, and it can be present in honeybees as well as the deforming virus in bumblebees, but we generally found it in higher prevalence and loads in bumblebees. So that's why I will qualify here as the bumblebee virus that can be shared across different bumblebee species and also into the honeybee, and sometimes even interact with the varroa mite. So back to the sampling design. So we've been collecting 6,000 samples, and this is the samples we've been screening for this virus in terms of presence, absence, and when it was present, we also quantify by qPCR the viral load in the positive samples. So we brought everything to the lab, we bisected it, and we used this 1/2 to run qPCR, and also later for RNA sequencing and viral discovery, and I'll talk about that at the end. And the idea was to link that with different factors, including the network. So this is what you've seen before. We used the plant pollinator networks to draw contact networks. And if we superimpose data on viruses, basically we are in this situation where we're collecting a lot of data and try to understand what's actually influencing the movements of diseases, as we did with these little apps. And the idea is to identify these factors and potentially to propose novel control strategies, especially in agricultural practices that can influence disease transmission. So these are the different factors that we measured and we used in our analysis. And we use farm type, so whether it's involved in the conservation program or not. And I would also mention that there are good farms or bad farms. The flower diversity, which is the data that we've been collecting on the farm when we were visiting and collecting. The landscape data, which here, it's basically around 2 kilometers around the farm. We measured an index of good habitats, which is based on which flower were present, which means mass flowering crops, but also these wildflower strips. And based on the literature, we give like a number of how good this area could be for pollinators. And we also use data on the composition of the bee community, and of course the contact network information to test the effect of niche overlap. And we also tested the position of some focal bee species on the transmission of diseases. And what we were expecting is that good farms, so HLS or the good farms that are involved in the program, would decrease the prevalence and loads of diseases, as well as good high plant diversity, good quality landscape, good diversity of insects. And while we expect niche overlap, when they do share the same food to boost disease transmission. So that's our expectation. So let's have a look to deforming virus. So deforming virus, what we found first, if we look at the colors here, and we found in red the good farms, the farms involved in the conservation program, to show a lower viral load for deforming virus across the different insects. Also here, it's not that clear, but we could also see that pattern in terms of prevalence of the disease. So we already have like a farm type effect. We can see that farms involved in conservation programs show like a lower viral load for deforming virus. So that was very interesting. And then we also looked at the effect of the bee community. And what we found is that pollinator diversity decreased viral load. And that means that in general, when it's more diverse in terms of species present there, we found less often we found a lower abundance of deforming virus. And accordingly here, we found the niche overlap when it increases to drop the prevalence of deforming virus, where this is the opposite of what we were expecting. Because here, when the bees are sharing their food, their forage, we found less of the virus. So it's more consistent with what we call disease dilution here. And although that was not what we were expecting, it actually makes sense, because that means potentially that the honeybees will share the disease, that they can carry very high loads to diseases that are less competent for that virus. And that's why we would find potentially a dilution of that virus. And then we compare this result with acute bee paralysis virus, which I recall it's a bumblebee virus, and there the main factor was the proportion of bumblebees in the community that had an impact on the prevalence, while the other factors had no effect. So here we have a big difference in response between these two viruses, which highlight how the epidemiology that can be different can lead to these results. And so what was interesting is that, especially for niche overlap, we were expecting the opposite result. We also look at the phylogenetic relationship between the different strains we had, because, as I said, we collected RNA from 6,000 samples, so we had access to the haplotypes of the species of the viruses in the different samples. And what we could see is that they do highlight two viruses with really different epidemiological history, and where we have a lot of mutation and a really recent history for deforming virus B, which is an emerging disease in the honeybee, where in the bumblebee here we have a more structured, especially geographically, populations of ABPV. But what is also very striking, and if you follow the colors here, we can see that these are multi-host viruses that can jump from different species of bumblebees and also into honeybees and circulate freely within this community of pollinators. So the conclusion of that second part is that white flower strips seems not to be a disease hub, at least when we look at the deforming virus. And there is no bird feeder effect, but rather we identify a disease dilution effect, where we found lower deforming virus prevalence when there is a high niche overlap. That means that bees are foraging on the same resource. We found a good nutrition effect and a good farm effect, where the conservation program had an effect on deforming virus prevalence on loads. And what I found interesting is that looking at different diseases, different epidemiology, really highlight that one change in agricultural practices will not affect all the diseases. And this is really something to keep in mind when we design, when we try to take action to tackle one disease. It may not have the same impact on other diseases that may also have important economic consequences on here on pollinators. Right, so then I'll finish with this last part on the factors that shaped the bee varroes. So varroes are obligate pathogens, and they replicate in host cells. They often co-evolve with their host, but they can jump into other hosts, and many viruses can be multi-host. And this is what I was showing previously with the phylogenetic tree between the bumblebees and honeybees. They are freely circulating. And the varroa are quite often very diverse phylogenetically, epidemiologically, and in terms of pathology as well. So this is what we wanted to explore, because in bee virology, we know very well the one that cause diseases, and there are many, but in the recent years, there was some interest to understand how particular these ones could be, and can we compare them to other diseases that are present and could be also very common, but do not lead to clear pathology. So a very diverse community, and this is what we wanted to dive in and try to understand the factors that change overall the varroes of bees. So again, same data set here, and because we had 6,000 samples, we didn't do into 6,000 different libraries, of course. So we had to pool samples, and we decided to go that way, to pool by species and by time point. So basically, we used the 3 first time points of our sampling scheme, and we had a pool of honeybees in April, in a pool of honeybees in June, and another one in August. We did the same for Bombus terrestris and Bombus lapidarius, while for the other 2 species, Bombus pascorum and hortorum, we had pools only in June and August, simply because these bee species emerge much later in the year. So this is when the queen will emerge for these 2 species, and the quill will emerge much earlier for Bombus terrestris and lapidarius. And then we follow a classic pipeline of virus discovery from the RNA of these pool of samples, and we dived into the varroes. So we found quite a lot of viruses, and what was first really striking is that we had quite a lot of plant viruses in there. So obviously, you understood we didn't wash the samples before. We really tried to get the overall varroa there. So a lot of plant viruses that were on the body from the pollen that they were collected. But we also found quite a lot of insect viruses, and including a lot of new viruses. And when we looked at this picture, we performed a clustering of this on these heat maps. And what we can see here, from the color pattern on species, we can see that the different species of bees do not share a big part of their varroa. And we know that some of them are transmitted from one species to the other. And we just showed you the data on deforming virus and acute bee virus. But overall, the varroas are quite separate. This is the honeybee, and we see that a lot of honeybee diseases are not present in others. And these are all the bumblebee species that show somehow a good separation of their varroa. And when we contrast that with what we observe with the plant viruses, here it was very interesting that the clustering analysis showed that there's varroas clustered by time point, and that makes sense. It follows the phenology of plants. This is when the bees were collecting the pollen and the viruses in the pollen that they were collecting at that time. And we can see it here. For example, we have the viruses from April, from spring, that are emerging together, and we have the prunus tree and the cherry tree viruses that are emerging at that season, and we can find it later in the season. So these 2 viruses type from insects and plants are not showing the same patterns in terms of clustering. But we know that all the viruses are not infectious. It doesn't mean because we see a virus in one sample that it's actually replicating and infecting. And because we had all this RNA, we also investigated what could be in the small RNAs, and will translate a potential detection from the immune system of the host. So through RNA interference, so the antiviral response of the host, we would expect when RNA virus is replicating and become double strand, it's recognized by this pathway, and through the risk complex, there will be a cleavage of the viral genomes into small bits of 21, 22 nucleotides. So we aimed at sequencing the small RNAs of these samples to identify which one were recognized by the immune response of the bee, and to have a hint of which species or which virus is replicating. And what we found with the Venn diagram is that roughly half of the viruses we found were replicating, while the other half were not replicating. So that was very interesting to see that, because there is a huge amount of data now from PCR that we identify this virus in this species. Where in the end, we never know if it's just acquired without replicating, or if it actually make the bee sick. So now, with this, we have a better idea, or at least a picture here of what was replicating in these samples that we've been collecting. And when we look into disease sharing and compare the data from the RNA-seq and the small RNAs, we can see that there is, especially if we compare in black the honeybees and in orange the bumblebees, so we have the bee species on top and the viruses on the bottom of these networks. When we compare these networks, we can see that there is a good share of viruses that we can find in both honeybees and bumblebees. But when we looked at the viruses that show a small RNA response, there is only a small fraction of them that are able, at least in our samples, right, to replicate into different species. Of course, we are aware that there are suppressors of RNAi in some viruses, and we cannot rule out some manage to escape that response. But overall, I think it's the picture is quite telling that in the end, there is not so many viruses that are truly multi-hosts. So finally, we try to understand what are the main drivers that shape the biomes of these bees. And we used three variables: the host genetic distance of these different bees using mitochondrial gene, and we generated a matrix from that alignment. We also generated a matrix from the niche overlap to identify which species had the closest ecological niche. So we had the host ecological distance matrix. And finally, we used the phenology, which is the time point when we collected the data, to identify what drives this virome composition. And what we find for the insect viruses is pretty clear. It's basically genetic distance. So it relates to this clustering analysis. Is the closer the phylogenetically two species are, they will share most of their viromes, right? And when we have distant species like honeybees and bumblebees, they will share less of their viruses. On the opposite, when we look at the plant virus composition in this virome, what we found is that we have an interaction between time points, so the phenology that explains why we found the virus at a certain time point, interaction with the niche overlap, and that's relate how the preference of the bee species for certain types of flowers and food will also be observable from the virus compositions here. Right, so in the end, what we learned here is that not all potential transmission are realized. So we did observation on which bees are gaining on which flower. So we had an idea of who could interact with whom. And in the end, we use the metatranscriptomes, the RNA-seq, to identify which viruses this was there, and we can tell that not all the viruses are spreading everywhere. And when we look at the different levels from the small RNA, we could tell that even if they transmit to a different species, they may not even replicate in this species they interact with. So that's the different layers where we can see these results, and that's why we also believe that it's quite important to dive into this level to really understand what are how the viruses there, to identify which viruses are actually truly multi-host, and to focus on these diseases that actually having could have an impact on pathology. So take-home messages here. It's important to link disease prevalence to relevant ecological questions here, especially when we want to take action here in the case of protecting bee health, and trying to find potential practices that will help reducing disease transmission. But what's also important is to explore the viral diversity and try to compare different viruses with different epidemiology and identify what are the change in practices could have in more than one species of pathogen. And again, as I just said, detection is not infection, and it's also important to always include that step when we are interested in disease transmission in species where it's also very difficult to tell whether they present a pathology from these infections. With that, I would like to thank a supervisor with whom we did that work, Lena Wilfert, which is now the head of the Institute of the Institute of Biology of the Institute of Regensburg in Germany. Toby, which did the field work and lab work with me, and Robin, another postdoc that were involved in that project. All the helpers on the field and collaborators, Claire and Mark, and of course you for your attention. Thank you very much.
Yes, thank you. Maybe it's not directly related to what you said, but it was kind of struck by the image of the wildflower strip that you showed, that there were many species that were actually not native in England, like the sunflower. Cecilia might be, but I'm not sure. So does the species composition of these wildflower strips also change something in either the connectivity of the networks or the pollinators that are attracted to it? It's a side question, but maybe interested to hear.
Yeah, thanks for these questions. And I originally wanted to test that, but unfortunately, we had no say on the composition of flowers. Of course, the farmers were completely free in designing or buying the mixes of seeds they use on their land. So we had a very different there, difficult to replicate anything there, very free. So I think that's an open question. I would say probably yes, because facilium that you mentioned, it's like a hive.
it attracts so many bees at the same spot. It's bad compared to other species where they are used when we are not that attractive. It's also difficult because based on what is available around, this is also one variable that will make the bees making their choice. So sometimes one plant that could be fairly attractive will not even compete with something that's even better. So there are all these variables to take into account. So I think that experiment was not designed for it, but definitely it's something to look into. Yeah. And to add on that, my colleagues at the University of Ulm were involved in motivating the farmers to use local flowers on field margin in the south of Germany, and that was also something that look into, and we don't have any data yet, but that would be very nice to, because there is a trend now to use local flowers and not use imported flowers.
Hello, thank you for nice talk. So I'm ignorant of this kind of wildflower strips, but are they all like coming like strips, like a long shape, elongated shape, or do they come in different shapes? And if you look at the effect of different shapes or sizes of the strips on bee and other insectivores?
They do come into different shape, and that's of course due to the landscape, some roads, streams, and so it could have an influence in terms of density. But generally, I would say the percentage of the field it takes, it's quite often the same, because this is all really well organized in terms of subsidies. So it's really clear what's the percentage of flowers they should use, and generally the minimum to get the subsidies, and use the rest for their own work, which is growing crop food. So probably there is an influence of this shape, and I think this is something that can be done also from mass flowering crops and the size of this resource. And yeah, so I would guess yes, but that's not something we could test here. The only thing we could see is that the percentage of good habitat, which is basically based on that, which is the proportion, didn't have any effect on the two viruses that we looked into.
Yeah, thank you very much for your talk, Vincent. I was wondering, so yeah, you didn't find any what you call the bird feeding effect, right? But it was at the interspecific levels between the hosts, like bumblebees. And I was wondering if you have any insight at the within species levels. For example, do you determine the colonies, for example, within the honeybees? And do you have a bird feeding effect at the within species level?
So we do have data on the colonies. So based on lights we used, and we know that bees from the same colonies will tend to share the same diseases. So there is this effect, and now there are. So I'm not part of it, but I know there are experimental work where we try to dive into this within species interaction, and this is more experimental, I would say, in semi-natural environment where we take flowers, we decide which flowers and the density and diversity of flowers, and we let bees. Generally, bumblebees, it's more easy to manage than honeybees, and we let them forage and see how the density of the flowers and also the state of the colony can influence then the spread of viruses. And I have colleagues doing this, showing that again, comparing a unicell and a virus, we could see different trends there. And it's also really linked to specific diseases that would take benefit of some variables in an experiment, that we could see something on that.
Just out of curiosity, how many different colonies do you observe on a given patch?
Well, that's a good question. I can't remember, and that was very high, because in the UK, also a rural area. So there is a lot of beekeepers. That was very high in terms of colonies nearby what we could actually detect. That was extremely valuable and 30 workers that we collected at each site. If I remember, we were around 8 on average, but it was quite valuable. Yeah.
How much do these different viruses share routes of transmission? And if they do, could this be used to establish a sort of expected value of the overlap with different values that you could get?
Can you reformulate? I'm not sure I understood.
Well, if they all have the same route of transmission,
yeah.
I would naively expect them to see a lot of mixed co-infections. While if they have very different routes of transmission, I would expect to see fewer co-infections. And actually, if you have data on the different routes of transmission, you could make a more quantitative argument and then try and see if what you observe matches this expectation or not, and if not, try to understand why.
We didn't look into this rate, but we know that we found more expected. So that's happened, and there is a trend now to focus on one specific bumblebee, which is Bombus lapida, used to check with the that could have actually rate of transmission and try to identify there whether they be health infected or through the foraging spectrum. So that's something that I have colleagues in the UK investigating. So that's Personally, I haven't been diving into that, but that's ideas that are emerging now.
I don't really have a lot of background. Do you therefore an increase in niche overlap with an increase in diversity of plant pollinators because you have higher density of bees?
Can you repeat that?
Sorry.
Very well.
Oh, I didn't know that. So you find that higher niche overlap is linked with lower prevalence. And so the increase in niche overlap is because of higher density of bees in higher diversity patches. So
the higher niche overlap was linked to the diversity of flowers there. So when there was a lower diversity of flowers, different bee species tend to forage on the same resources that were available. And when there are more flowers, they tend to not share the same resources, so to forage on their own preferred flowers, which is generally defined by their own physiological needs for themselves and the colony.
Okay, and so the dilution happens because you have more number.
Exactly. So we had so basically what we see is that when there is lower diversity of flowers, we have higher niche overlaps, and that leads to dilution. And that's not a result that we expected, but it seems to be verified in other bee diseases in completely independent work.
Okay, thank you.
Thank you for the talk. I had a follow-up question. So how does the dilution effect work? Because I'm not familiar. Does it mean that different viruses are in competition between them, or?
So at the virus level, let's say there are landscape of different hosts, and so these hosts represent places where it's more benefic than others. And here, in the case of the farming virus, for example, it can grow very well at very high loads and transmit very well between honeybees, where in bumblebees it will be there, but generally quite at lower level. potentially because the immune system is more efficient against the forming virus, or because of coevolution and is not adapted to bumblebees. What we see from the small RNA, for example, is that it replicates very well in honeybees, where we couldn't find any siRNA reads in bumblebees. So although we could find it in the transcriptomes. So it was present in bumblebees, and we know from qPCR that it was there at lower level, and it was not replicating very well, obviously. So this is what it means. When they share the same resources, basically the honeybees will spit the virus on the flowers, will be collected by other bees that are less competent for that virus and will represent, let's say, a dead end landscape and a dead end for transmission potentially. Yes, thank you.
Sorry, about the dilution effect. It's very interesting. So, but I just make sure I understood correctly. You found dilution effect for the first virus, or what that one, deforming virus, and then you said you didn't find it in
For
the other one. But you said that if the bumblebee dense proportion of them bumblebee is bigger, then the acute virus prevalence is higher. That means that they like between different bumblebee species of, I don't know, maybe you looked at only bumblebees or like honeybees and maybe butterflies and beetles and so on. So could we expect a dilution effect among these very diverse, different organisms, like different butterflies?
Yes, of course. Yeah, that's a very good question. Indeed, we would expect that. The design of our work in the end, because we were collecting on these patches of flowers, we mainly had bees, and in 90% of them was social bees like bumblebees and honeybees. We have very little other species of insects in the end because of the choice of flowers that were used. So we couldn't really test that. We had in the end we had 2% of insects that were not among the species I was talking about today. So we could see in other bees these viruses, but that was more anecdotal given the numbers. And then when we talk about flies, we still had quite a few flies there. The virum were even different. So yes, in the end, we would expect, if there is a good ratio of flies against bees in terms of pollination, we would expect also the flies to collect the virus as well through the nectar and to not be a good host, because we can see that in the sequencing in the end. But that, I would say, to make sure dilutions happen, one would actually check whether the flies are able to catch the virus, because the moth parts are not exactly the same. And also the overlap sometimes is not very good given the type of flowers. So I would say more work specifically, I would say in natural habitat there would be interesting to check on other groups of insects.
So thank you very much. Time to finish. Thank you for being dispersed in the first seminar. It was really nice, and thank you all for being here. See you next week.